So my lab, I just started at Mount Sinai, and we're really interested in how we link memories across time, very related to what Tim was talking to you guys about. And what I think is amazing is that the brain is able to form memories across a lifetime and then store it, organize it somehow such that you can efficiently and effectively recall and mentally time travel to the past. So for example, we'll time travel together a little bit. I can think back to one of my fondest days, and that's the day I got married. And I might think back to that day when my dad walked me down the aisle, promising me that he would never cry, but he was moved to tears by the man I was about to marry. And recalling that memory might then trigger a subsequent memory, such as later that day when my husband and I get to toast our family and friends and live happily ever after, no more wedding planning. And thinking about that memory might then trigger a subsequent memory, such as when our guests fully took advantage of the open bar at our wedding reception. And somehow my brain is able to temporally relate all of these events together such that I can recall them in these kinds of sequences and know they happen close in time, and separate them from other events, such as here, my husband and I are toasting to a different occasion in Napa Valley. So the question that I'm really interested is how is it that our brain is able to temporally relate these events across time and distinguish them from other events further in time? Now, I'm going to summarize some published work that was published in 2016 to kind of set the stage for what my lab is looking at now. So in the published work, what we showed was that mice explored a brand new context. It's like a new world to them, episodic memory. That memory gets encoded in a sparse population of neurons. And those neurons actually, or what we call ensemble cells, translate increase in excitability over across the time. So in our paper, we show across 5 hours, those neurons have higher excitability than their neighboring neurons. And others have shown that it takes days and comes back to basal levels. And what we showed was that when those neurons are high and excitable, they're more primed and ready to go, more ready to capture the next memory. If we then present a different context to them, they now are more likely to overlap in the neural representation, and that this is how we think we link memories close in time, and that when the animals then go to a third world days later, then the representation is encoded in a different group of cells, and that by just putting the context closer in time, that they're going to share an increased overlapping representation, and this is how such that when the animal thinks of this blue box, it's going to trigger then the recall of this red box, and much less likely of this green box. Okay, so what we showed was then, in this paper is that essentially these kind of neutral memories of animals exploring novel contexts, within a day, the memories are much more likely to be linked, and they're separated across days. So that makes sense, right? We don't want to overlink. We don't want to link everything with the world. We want to link things that happen close in time and separate them that happen distinct in time. Now, one of the questions that my lab is currently trying to answer is, what happens if you make something really traumatic? And does that alter the window of memory linking? And so the experiments I'm going to show are pretty new. Our lab just started, and they're primarily done by a fantastic research associate, Brandon Wei, who came with me from LA, and Phil Dong, who's my graduate student. Okay, so what they want to test this idea, we know that memories are encoded in a sparse population of cells, and here we're looking at the hippocampus. But what happens if you make memory really traumatic, right? You give them really strong foot shock. Does that now alter the window of memory linking, such that maybe what happens is that the excitability of these cells are maintained for a much longer period of time, such that now they're going to link this fearful experience with other safe experience that happens in the future, and that this fear is going to transfer to safe context, and that this would extend prospective linking. Alternatively, you could also imagine that maybe the fear of that blue context gets transferred to events that happened before, right? So we're testing, is there a symmetry in memory linking with retrospective and prospective, and what happens when you add negative valence? Okay, so our approach to this is using a miniature microscope system. So many of the speakers have already talked about in vivo calcium imaging. When I was at UCLA as a postdoc with Alcino Silva, in collaboration with Payman Ghoshani's lab and Baljeet Cox's lab, we developed an open source miniature microscope. And so basically, just a few plastic pieces, an LED, a little camera chip from your cell phone, and we got this puppy for under $1,000. And I was told none of my videos work anymore, so what you would see on this blank screen is an animal wearing this miniscope, exploring the context, how boop, 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 boop are going off, and are encoding their context. Okay, so this is the imaging study. The animals are going to explore 2 different boxes, and there's different temporal intervals that are separating these 2 boxes, and we image the cells that are active in each of these contexts, representing these contexts. And then we look at what is the neural overlap between the red box and the representation of the blue box. And what we find, similar to what we published, is that at 5 hours, there's an increase in this neural overlap, and that it decreases across days. So previously, I said, well, thinking about one through this neural overlap is going to trigger the memory of the next one. So to probe the behavioral memory, what we did was the same experiment as the imaging, except now we bring them back to that blue box, and we update that blue box with the shock, so that if they were to go back into that blue box, they would remember being shocked there, and they would freeze because they're scared. But putting them in a different context usually does not elicit freezing. So what we saw when we put them back in that red box, remember, they've never been shocked in that red box, is that at 5 hours, when there's higher neural overlap between the 2 representations at initial encoding, they also show more freezing, this transfer of fear to this red box. And this is temporally specific. It's not that they just freeze to all other boxes. As you see by the next day, when there's a decrease in the neural overlap, there's also a decrease in that transfer of fear. Okay, so this is for when 2 contexts are neutral, right? Similar to our published results. So then we ask, what happens when we alter valence? So to test prospective linking, what we can do then, the first time they go into that blue box, we give them that shock. And then we ask, are they going to transfer that fear to a novel context that they see in the future across these time points? And then we put them back in that red box to test what is that transfer of fear. Alternatively, we can test retrospective linking. So in this case, we shock in the blue box, but they saw a red context that was neutral hours or days ago, and then we test them back in that red box to see if there's that transfer of fear. So what we see for prospective linking is actually very similar to what we see for 2 neutral events, such that we see there's a high transfer of fear when the 2 experiences are separated by 5 hours, but it decreases by the next day. Interestingly, for retrospective linking, we saw that the mice were transferring that fear to save context that they've seen even up to 2 days ago, right? And this is actually totally opposite of what I would have predicted, and based upon what we published, this is not what we would have expected. However, then we might think, why would you want to retrospectively link? So I was recently trying to get to Utah. I just moved to New York. I'm a native Californian, and I got stuck at JFK Airport during the bomb cyclone. And that was pretty traumatic for me, especially as a Californian. What do I do, JFK, during this bomb cyclone? Can't get to Utah. And I was thinking back, goodness, what could I have done to prevent this? And I saw on my phone 2 days ago, my colleague Mark Baxter had texted me and said, hey, there's a storm coming to New York. Maybe you want to switch your flights. But I didn't do that. And so maybe in reflecting upon the past, I can now make better decisions about the future, which is always listen to Mark Baxter. And maybe the reason we want to retrospectively link is because the past predicts the future. And so if you have a really negative experience, you might want to look further back in time for any predictors of that future experience. Okay, so previously I told you that what we found in our published work is that what links memories together is this overlapping population of neurons. So we decided to just look to see, is there an increase in the overlap of neurons during this, when it's initially aversive in this 2-day group compared to when it's initially neutral. And so this is just to remind you of this. I've already shown you this data, that everything is the same except they're either having this blue box and experience a shock during the initial encoding, or we update it later. And we see that when we initially make it aversive, we see this nice transfer of fear, which we don't see for initially neutral. So those are the 2 groups that we took, and we started doing imaging experiments with them. And here we're recording from dorsal C1 and hippocampus using calcium imaging. And throughout the entire experiment, and so here is the neutral group. They explore these 2 boxes. They get updated later. Now they're tested in one of 3 boxes. And here we have a novel box just to test for generalization. And so now we're looking at the overlapping ensemble between the red and the blue. And what we see is that the overlap between this neutral group and the overlap between this aversive between the red and the blue box is no different at encoding. Okay, well, that's not that surprising. So what happens when they go to retrieval where we see the transfer of fear? What we find is that sometime between encoding and retrieval, these 2 representations change, and that there is an increase in the overlap. There is an increase in the overlap in the aversive group during this retrieval process, maybe reflecting this transfer of fear now to this red box. Now, my very smart graduate student said, well, Denise, maybe it's really just the behavior, because in this group, they're freezing here, and the behavior is driving the hippocampal activity, causing the same cells to fire, where the behavior here is very different. So then we looked at the neural overlap between the shocked context and this novel context in which they don't freeze in, and we find that there's no difference in the neural overlap. So it's not that they're just increasing or generalizing this overlap to any context. And we also see that there's no difference between the overlap between the red context and this novel context. So this suggests that when something is happening between encoding and retrieval, and this is kind of where we're at right now, and we don't really know, so I'm hoping to crowdsource really smart people in the audience to tell me what is actually going on. So Tim did a really nice job explaining replay. So maybe there's some offline co-reactivation of the red box and blue box. Maybe this happens during sleep. And my graduate work would actually suggest that maybe REM sleep is a state in which we integrate very recent experiences with past experience through this reactivation of these memories. Alternatively, it could be active rumination. In the example I gave you about being stuck at the JFK airport, I was actively trying to think, how could I prevent this in the future? So we don't know. So the first thing we want to do, though, is start to look to see what is the activity going on in this offline phase. And so we've been using this wired scope. This is a published version. So also in collaboration with labs at UCLA and Tristan Shuman, we've developed the wire-free version. So this is unpublished, and Tristan Shuman will be presenting a lot more of this data tonight at the poster session. And so with the wire-free, we can now image the activity in truly freely behaving in their home cage during offline periods. The data gets logged onto a micro SD card, and it runs on a lithium-ion battery, so it's completely untethered. So this is not the video that should be showing up, but if the videos work, you would see a mouse running around untethered, but you know, that's all right. Okay, so using this tool, then we want to go in and look at these different stages during this offline period and ask, how are these representations being linked together? So in summary, I showed you that neutral memories within a day are linked through this overlapping ensemble, and by having a really negative, adding negative valence, it actually extends the memory window, but retrospectively, and we think also mediated by this overlapping ensemble. So I just really want to highlight Phil and Brandon, who did really all the studies, and my new lab. This is us at Mount Sinai. I also want to thank my amazing team Miniscope from UCLA, and also Tristan at Mount Sinai. I have to give a shout out to Daniel Aharoni, who is the brilliant physicist who developed all of these tools that we're sharing through open source platforms. And thank you for your attention.
We have time for 1 or 2 questions.
So here we're recording between 400 to 600 on average that are active in in each context exploration. But we have access to over 1,000 cells, and different cells will come in different times across the context.
I can repeat it.
Hi.
Well, hold on, just 1 second. Sorry, Anne. Do you want to come up to a microphone?
So I was just interested in you're giving us a notion of quantities, how many cells in a given ensemble, they must differ in their numbers, and what constitutes overlap, and
Right.
is that
Yeah, so that's a really great question. So using this technique, we have access in hippocampus. Our field of view, we have access of over 1,000 cells. We don't know exactly because with calcium imaging, we don't see silent cells. We only see the ones that fire. And so what we do is we put them through 10 different contacts, and then we count all of those cells. So we estimate that on average, between 400 to 600 cells represent each context out of 1,000 to 1,500 cells. This is consistent with other tetra studies and in vivo studies. So we've also we wanted to know how much activity does a cell need to have to be part of this context. So we've looked at that, and it seems that what that does is it just changed the absolute amount of overlap, but then it doesn't change the relative difference across time. And we can chat more about kind of the concrete numbers later. Thank you.